I only have one complaint, which is that if I forget to launch the quiz and I say there's a quiz, somebody should tell me. When it says waiting for instructor, what does it say? Waiting for instructor to start. Tell me, please. Don't just sit there. Okay, so today, and maybe into a little bit of our next class period, we're going to talk about the chapter that you read about for today and that the Socratic exercise is on, epigenetics. All epigenetics is a fancy way of regulating gene expression. So when we talk about gene expression, what do I mean when I say gene expression? What does the term gene expression mean? Yeah, when genes are being transcribed and translated, the last two things we've been talking about in class. Okay. So this is yet another mechanism of control of whether or not a gene gets turned into a protein. It's not just does the gene get transcribed and does the transcript get translated. We have to know something about epigenetics as well. So these are the goals that I have for you for this class. One of which is simply to define what epigenetics is. How you might figure out whether or not a trait is subject to control by epigenetics, and then being able to predict, if I tell you something about the epigenetics of a gene, predict whether that gene is turned on or turned off. Is it transcribed and translated, or is it not? So those are the outcomes that I'd like. Any questions or concerns? So some of you have already obviously been looking ahead in the slides, which I love. Good. Thank you for your feedback on Socratic. Keep it coming. So epigenetics, all it is talking about chemical modifications of DNA. So all these types of molecules that are in the Socratic quiz get modified. DNA gets modified, RNA gets chemically modified, proteins get chemically modified. The epigenetics talks about DNA and proteins specifically. First, we're going to talk about DNA. So what are the types of chemical modifications that happen to DNA? Almost everybody got this on the Socratic quiz so far. So what chemical group can get added to DNA? Methyl groups. Right. So CH3. So this is not mutation. That's the critical point. That epigenetics has nothing to do with changes to the DNA sequence. It all has to do with additions of chemicals on top of the DNA sequence. And somehow those chemical modifications of DNA, enzymes come along and stick a methyl group here, a methyl group there. Somehow that changes whether or not a gene gets turned on or not. And the reason this is called epigenetics is epi is a Greek prefix that means on top of or above. So literally, epigenetics is the study of things that happen on top of DNA. Chemical modifications on the DNA molecules that change how our genes get turned on and turned off. So this slide is really just a sort of a general overview of what epigenetics is all about and what we're going to be talking about today. Part of it is, as you said, methylation of DNA itself. Right, that's one of the chemical modifications we look at. The other one is the modification of proteins. So in which proteins are we talking about in this case? It's proteins associated with the DNA, which is why those proteins can epigenetically control gene expression. I heard one person say it. What are the proteins that are up here? Histones. Chromatin. We talked earlier in the term about when chromatin structure is open, like here. Then proteins can access the DNA, the nucleosomes, the histone proteins that are grouped together in groups of 8. The nucleosomes are spread far apart. The DNA in between is open and accessible for transcription factors to come bind to, recruit RNA polymerase, start transcription. And so those histone proteins get chemical modifications as well. And that can control, among other things, whether or not the nucleosomes are spread far apart or whether or not they're all packed together, like shown on the left here. So this compact structure would be repressive for transcription. The gene turned off, in this situation, the gene would be turned on, expressed in this situation. So that's the brief overview of where we're headed. So we're going to learn about what are the chemical modifications and how do they control gene expression. We'll look at a couple of case studies. So DNA methylation, as you read about, happens primarily at CG nucleotides. They're also called CpG because the P is for the phosphate that's in between the C and the G. I don't know why they bother to stick that P in the term, but there it is. So it's when a C and a G are next to each other in the genome, the cytosine, the C, can be methylated by enzymes that add methyl groups onto cytosines. What's special about CG? I don't think the chapter mentioned this really. Why CG? More hydrogen bonds. That's a good guess. I don't know if anybody really knows why CG. What's on the other strand opposite a CG dinucleotide? A CG going the opposite direction. So guess what? There's another C that can be methylated. So at spots where there are CG dinucleotides, both strands get methylated. The parental strand and the newly synthesized strand of DNA both get methylated. That's really important because these epigenetic marks are heritable. That means that you pass on your chemical modifications of your DNA molecules to your kids. Because when DNA is synthesized, when the two strands separate and the new strands are added, synthesized, these same enzymes that add the methyl groups to our DNA come back along and remethylate the newly synthesized strand of DNA. And when those chromosomes get packaged into gametes and the gametes fertilize and produce the next generation of kids, they inherit our methyl groups, essentially. So DNA, tell your friends, next time you're at a bar quiz or something like that, a pub quiz, if it comes up, DNA is not the only hereditary information. It's not just the sequence of your chromosomes that's hereditary, but the chemical modifications on top of your DNA can also be inherited by your kids. So by the end of the two or three case studies we're going to talk about, I want you to realize that the choices you make and your lifestyle decisions and your environment impacts your kids through you, if you decide to have kids. If not, go wild. Even if you do want to have kids, go wild. You're in college. Go for it. So, wait, I'm in college. Maybe I'll go wild too. This is just the difference between cytosine and methyl-cytosine. So, big deal. There's a methyl group that gets added to cytosine. What does this do to gene expression? Why is it that by itself, the addition of a simple moiety, does anybody talk about moieties in organic chemistry anymore? Okay, never mind. I see heads shaking, so I'm not going to use that term anymore. Chemical groups. Why does this change gene expression? Changes the shape of the nucleotide. Thoughts? Well, here's an example, potentially. So let's say there are promoter sequences where those transcription factors bind. This is a gene. Transcription factor is trying to bind to the promoter, recruit RNA polymerase, and cause transcription. Except, what does it look like is happening with methylation of these CG dinucleotides? Pardon? So this transcription factor is physically blocked by these additional chemical groups from recognizing the sequence that it's trying to bind to. So the chemical modification, this is one way that methylation can change gene expression. You're changing the shape of the DNA, and sometimes, DNA or proteins that are trying to recognize those DNA sequences don't recognize the DNA sequence anymore. Those nucleotides are not C's and G's anymore, they're methyl C's. And the protein doesn't recognize that as a sequence that it binds to. So methylation can turn gene expression off. There are other ways in which methylation can actually activate gene expression. So you can't predict bottom line, can't predict by methylation whether a gene is turned on or turned off. Most often, though, methylation of promoters is related to inactivation of gene expression for this reason. Any questions at this point? So one of the ways our cells regulate whether or not genes are turned on or turned off is we have enzymes in each of our cells that go around our DNA methylating C's in CG dinucleotides. Not all of them, though. Yep.
How does those proteins or enzymes randomly choose arbitrarily? Is it like specific ones like, oh, I don't want this gene, so I'm going to list
Right. So the question is, how do the enzymes that add these methyl groups know where to put them? One of the enzymes you read about in the chapter are maintenance. So the enzymes are called methyltransferases. Guess why? Yeah. Maintenance methyltransferases transfer methyl groups on those bits of DNA that are hemimethylated. That is, you could imagine there's an original strand of DNA and the newly replicated. So which C is going to have a methyl group on it right after replication? The original or the newly replicated one? So if the original one had a methyl group, use a red asterisk. These maintenance methyltransferases read the DNA to find the hemimethylated sites. This is hemi, half methylated. One strand is methylated, the other one hasn't been yet. And so every time that enzyme sees something that looks like this, it adds the methyl group, oops, sorry, not to the G, but to that C. That's why these are heritable, and that's how the DNA in the cell knows where to methylate. It's based on what was methylated on the original strand of DNA. Those marks just remain. The enzymes keep them, they just keep adding them to the same sites on the daughter strands that are after synthesis. Now that's not to say that new methyl groups can't be added, so that does happen. And sometimes that's by environment. So there are chemical modifications, can be addition of methyl groups that doesn't have to do with the enzymes doing it. So your exposure to chemicals in the air, for example, if they get into your nucleus, sometimes your Cs get methylated not by the enzymes that are trying to do that, but by other processes, just chemical processes. The histones, as we talked about, are the other molecules that are deeply involved in epigenetics. So chemical modifications of the proteins that DNA wraps around can regulate gene expression. This is a bit more complicated than DNA methylation, only because there are a lot more types of chemical modifications that happen to these proteins, the histones. Two of the many chemical groups that can be added are acetyl groups and also methyl groups again. So here's just a brief reminder about chromatin structure. The one new thing that I want you to focus on in this diagram simply is that the histones don't, in reality, look like nice, compact, tight balls of protein. Their N-termini, which are called tails here, the amino termini, the starts of the proteins all hang out into space. So they don't adopt particular conformations when the DNA is wrapped around them. The N-terminal tails just hang out. And that makes them subject to this chemical modification. Epigenetic marks get added to the histones, specifically on their tails. So not in here in the body of the histones, but rather at their ends, the ends of the proteins. So DNA gets methylated on C's in CG dinucleotides. Histones get methylated, acetylated, and a bunch of other things on their N-terminal tails. This was all the rage in genetics and biology about a decade and a half ago, when a couple of researchers, well, many researchers, discovered the histone code. So you've learned A's, C's, T's, and G's for DNA sequence. A's, U's, G's, and C's for RNA sequence, the 20 amino acids, and now here's a fourth level of understanding of how our genomes work. How histones are modified. What sorts of groups are put on which parts of the histone proteins. So for example here, this is saying that there's a serine. It's serine 1. It's the very N-terminal end, the first amino acid in histone H4 can get phosphorylated. That's that P. So that's a chemical modification to that amino acid that occurs on histone 4 at its first, histone 4 at its first amino acid. There are other parts of histone 4 in its N-terminal tail that get acetylated. There are some amino acids that can get acetylated or methylated, and so on. So this is a painstaking amount of work for the scientists to figure out which histone is at H2, H3, or H4, and which specific amino acid is it that gets these chemical marks. The table below is from Wikipedia, so you've all got access to it. I took it from a highly scientific source. But that's because this is really context dependent. What a particular chemical modification of a particular amino acid in a particular histone, H2A, H2B, H3, or H4, is really dependent on the specific case. So again, it's hard to predict for a specific gene what one of these marks does. So that's why I like this table, because it gives you the in general. These are the broad patterns. What's H3K14? Can somebody unpack that nomenclature for us? What do you think that means? It's just an abbreviation. So what's H3? It's a histone. Okay. So that means histone H3. What's the K14? It's a lysine, K. It's the 14th amino acid in histone H3. So that's just saying H3K14, histone 3, it's lysine at 14th position, reading from left to right, can be acetylated, and when it's acetylated, that tends to activate gene transcription. That's how to read this table. So you'll see there are a lot of different amino acids, but which ones are the ones that always get modified? It doesn't matter which histone you're looking at. What sort of amino acid is the one that usually gets modified, according to this table? It's all H something, K something, right? It's a histone. It's always a lysine. It's always Ks. Or at least it tends to be. There are other amino acids that can be modified as well. But the ones that are really important in regulating gene expression are lysines, and what happens to them. Do they get acetyl groups, methyl groups, and a few other types, which aren't shown in this table. So DNA gets methylated, histones get acetylated, methylated, and other things, usually on lysines, any of the histones. So that's the factual stuff. That's what I hope you took out of the reading. Those are the highlights. So how did a bird get in here? Well, that's a good question, but I'll try to ignore the bird flying around in the room for a little while. Unless it's going to go in front of the projector, in which case, I don't know what to do. Do we have any animal biologists in the lab? Poor bird. Okay. I'm just so easily distracted. Who do you call when an animal invades your classroom? A non-human animal. Facilities? Okay. So let's get back to the biology. We'll ignore the organismal biology. Okay, I'm going to try to control myself. How do these chemical marks control chromatin structure? So the methylation, all I want you to take away from this is not mechanism, not how this happens, but methylation tends to cause the nucleosomes to compact together, as is shown up here. And acetylation of the histone tails tends to cause nucleosomes to be spread apart. So that's how that table comes up with those general trends. That if methylation of histones happens, that usually causes compaction of the chromatin structure. That will turn on or turn off gene expression. If the methylation, compaction of nucleosomes turns off gene expression. If on those same lysines, instead of methylation, you acetylate them, then the histones, the nucleosomes spread apart, you get genes turned on. So it's kind of like a switch. You've got a lysine, it's either methylated or it's acetylated. If it's methylated, the gene's turned off. If they're acetylated, generally the gene's turned on. So here we go, case studies in epigenetics. Let's see if everybody got, so let's see, fur color, asthma, twin studies, all sorts of good things. The bird's trying to tell us where it's from. Okay, a lot of you talk about fur color in the Socrative quiz. Maybe you're referring to the process of X chromosome inactivation. I didn't ask for details. So instead of birds, let's think about cats instead. I wish I had a cat, because maybe the cat would get rid of the bird. I'm just kidding. Sort of. So here's a calico cat. Why does it have this mottled fur coloration? Okay, if the bird starts dive bombing you, then you're, I don't know what to do. If we turn, there are two alleles of the coat color gene in cats. And like human color vision, coat color alleles are X-linked. So what happens if I turn off one of these chromosomes? Capital OR makes orange fur color. This other allele, lowercase or, doesn't make orange, it causes black pigmentation. So X chromosome inactivation simply means in females, we've talked about this briefly earlier in the term, in females, one of the two chromosomes gets permanently inactivated. And depending if you're a female cat, one of your X chromosomes gets turned on or off. If you're heterozygous, that's fine. If you're heterozygous, as long as it's not distracting us anymore, if you're heterozygous, like these females, then which chromosome gets inactivated, which we'll call X sub I for the inactivated X chromosome? That means that some patches of the cat's coat are orange. Those cells are the ones that have the X2 chromosome, and the one with the black pigmentation gene turned off. The cells where there are patches of black are the ones where the orange, the version of the X chromosome where the orange gene was turned off through X chromosome inactivation, are located. So the real question is, why isn't every single cell of the cat a different color? Why are the colors in clumps on the body? Rather than every single cell, which we wouldn't see, we would just see a single colored kind of orange and black cat. Why is it in patches on the body? Because you could imagine the case where every single cell is either black or orange, but they're all spread out. So every other cell is black, every other cell produces orange pigment. You have a cat that kind of looks like one color. Why is it in patches? We'll get to that in a second. I've got a short video to show. It turns out, like you might predict from what we've already talked about with methylation and acetylation, the inactivated X chromosome has those epigenetic marks that we usually associate with inactive genes. So you can refer back to the table later on your own time. Does low histone acetylation usually mean inactivation? Does low methylation usually mean inactivation? But in general, the X chromosome uses epigenetic marks. The inactive X uses epigenetic marks to turn itself off. Okay. So quick video. If you take anything away from this, try to figure out why in cats or in any animal that has X chromosome inactivation, why there are blobs or blocks of color on the body. Okay, so while the sound was working previously, what this is, do you see anything at all? Okay, tell you what I'll do. I will put a link to the movie on Blackboard. Technology failure. So anyway, what this movie says is that what happens during cell division. So imagine that you're a single-celled gamete, you've just had fertilization happen. You become a 2-cell embryo. Each of those cells divide, you become a 4-cell embryo, 8-cell embryo, and so forth. There's a particular point in development, early in development, when X chromosome inactivation happens. Randomly, that's the really critical part. Randomly, you get one or the other X chromosome. Your X from dad, let's say, is blue. Your X that you get from your mom is red. So at some point early in development, each of those cells independently chooses one X chromosome to shut off. So you'll have some chromosomes, like I'm filling in red, where mom's X chromosome is still active. Dad's has been randomly inactivated through epigenetics. And other of those cells will randomly have dad's X chromosome be the active X chromosome, the one that transcribes and translates its genes. So what happens over time as this organism continues to develop? Let's see, this is the 8-cell stage. So I'm speaking very generically now, what fraction of the adult body will that one blue cell create? There are 8 cells at this stage. They're all going to contribute to the adult body through cell division and differentiation. So that one cell is going to produce about 1/8 of the total body. And so will that red cell, I'm speaking very generically now, it's not necessarily exactly 1/8 of the body. But the point is that early in development, those 8 cells at this stage are going to eventually, through cell division and cell division and cell division and cell division, produce all of your cells. So this is why those female cats have blotches or blocks of color on their bodies, because all of those cells, let's say that have orange fur, all derived from a single cell early in development that turned off the X chromosome that has the black pigmentation gene. So that portion of that cat came from that single cell at the point in development when the X chromosome got turned off. In the video better, but in sum, every female body is essentially a mosaic of parts that have mom's X chromosome turned on and parts that have dad's X chromosome turned on. You can see this in a number of ways. In cats, you see it by coat color, and in other organisms, you see it by coat color. This also has some interesting forensic implications, potentially. Different parts of a female's body in trace DNA will be the same, but the epigenetic marks that are on trace DNA found at crime scenes might look like they come from 2 different people, when really it's just that skin cells from a cheek might have certain epigenetic marks if you're a woman, and skin cells from your toe might have totally different epigenetic marks because the X chromosomes that have been inactivated in different parts of the body are different. So what does cause the distribution and the size of the groups of cells that are all derived from that one chromosome that turned off one X chromosome at random? What would it look like if you were a 2-cell embryo and the X chromosome inactivation decision was made? What would you predict a female cat that was tortoiseshell would look like if at the 2-cell stage, each cell decides which X chromosome to turn off? Could be half one color and half the other, right? Because there are 2 cells. Say one turns off the black allele of the X chromosome and the other one turns off the orange allele. So you get a half this way, this way, this way. We don't know. But yeah, half the cat would be orange, half the cat would be black. The farther you get into cell division, the more cell divisions there have been before X chromosome inactivation happens means those blocks of color get smaller and smaller and smaller because you're talking about an embryo that has more and more and more cells. So that's X chromosome inactivation. Cells randomly turn off in females one X chromosome. That happens through epigenetic marks. So I'm here to say that there is no gay gene, despite a lot of work by a lot of geneticists, because of course it's a fascinating topic to a lot of people, and a lot of people want to know, is it nature or is it nurture? It's whether you're homosexual or heterosexual due to your genes or due to your experiences or a combination of both. Some really famous high-profile geneticists have made the simple point that if there was one single gene that caused homosexuality or heterosexuality, depending on which perspective you want to take, we would have found it by now. There have been way too many studies for there to just be a single gene that controls it. So that's certainly not the case at this point. But that doesn't stop geneticists from wondering, could it be multiple genes, the activity, all of which together contribute to sexual orientation? Or might there be epigenetic factors? So that would incorporate some of the nature plus nurture. Your DNA plays a role, but also those chemical modifications to your DNA might also play a role. So I'm here just to simply highlight a couple of studies that introduce and maybe slightly better define this topic. So here's a couple of interesting research examples that I'm not going to belabor in detail, but some of the early studies that got people thinking about whether or not there's a genetic basis to sexual orientation. First, the birth order effect. The more older brothers you have, the more likely you are to be homosexual. Right? So this study back in 2001 found that there's about a 20% additional chance for every older brother you have of being a homosexual man. Is that nature or nurture? Something to do with pregnancy. So I encourage you to actually go look at this study. It's really interesting. So yeah, what story would you tell about? Why does it matter how many older brothers you have? Some of the stories that are told are about mom's immune system, and that maybe over time mom's immune system builds up antibodies against male-related something that has to do with carrying so many male fetuses, and that maybe that somehow affects sexual orientation. That's total what we call a hand-waving argument. Look over here, I know what I'm talking about. Right? The hand-waving argument. So we don't know why this is the case. There's just a correlation. It's a pretty mild correlation too. Then we've got the twin studies, which are maybe a little bit more revealing, because twins, as we've discussed, if they're fraternal twins, are identical in DNA sequence or almost identical. Right? Maybe there are a few genetic differences. So what's the prediction? What do you think? You got 2 twins. What's the question you want to ask if we're studying sexual orientation? What's your prediction? If I tell you twin 1 is homosexual, what's your prediction about twin 2? You think he'd be
most likely to be homosexual as well.
if
Fraternal.
Yeah, if they're fraternal twins and twin 1 is homosexual, then the prediction would be twin 2 is homosexual, because if genetics controls this trait, if they're genetically identical, they should have the same traits, right? But there's only a 20% chance of that correlation. So identical twins, for the twins they studied, about a fifth of them, both twins had the same. If one twin is homosexual, the other one was as well. Can you think of reasons why this sort of research might be faulty or flawed? What questions have you been thinking about as you've been hearing me blather on about homosexuality in twin studies?
If they're twins, they'd be raised in the same environment, so if it is actually nurtured, then it would enhance that.
That's a great point. So if we're doing twin studies, yes, the twins are genetically similar, but if they were raised in the same environment, like the same house by the same parents, etc., then both their genetics and their nature and nurture are the same, or very similar. So how do we disentangle those? Often twin studies nowadays, is that a word anybody uses anymore? Currently, a lot of twin studies take place with twins that have been separated since birth to try to control for that. But, fabulous point. What else about this sort of research is difficult to analyze or assess? What's the phenotype that they're studying? We got the genotype, that's their DNA sequence.
Well, it's the homosexuality, but
Right?
for what about the people who are heterosexual for a long time, and then they say, oh, now I'm homosexual, but then like later in life, they go back to being heterosexual, so how do you classify those people?
Exactly. So at what point, right, this is important not only philosophically, but also biologically. It makes a really important point. How do you measure the phenotype you're studying? When you're looking at DNA sequence, it's obvious. You've got 4 letters, you know the sequence. What about a phenotype like sexual orientation? At what age do you ask participants in these studies, what is your current sexual orientation? Do you go back to the participants later and ask, hey, have you changed your mind? If that's a thing, I'm not saying it is. Have you changed? Right? Or is that trait static across your particular entire life? So these sorts of studies, when you're talking about behaviors, are really difficult. How do you define the phenotype of homosexuality? Is it how you self-identify on a survey? Or is there something else we can measure to ask, to classify which twin is homosexual and which twin is heterosexual, if there is such a thing at all? So all I'm trying to do is implant a sense that you need to be really critical and maybe even skeptical when you hear about these sorts of studies in media especially. Because there are a lot of important questions to ask about how exactly was the study done before you start drawing conclusions. And this will summarize that point very nicely, I think. So a little bit more into this case study. We'll probably pick up the other one next class. But to finish this one off, about a month ago, there was a ton, you might have seen some of it, there was a ton of popular press coverage on one presentation at the American Society of Human Genetics conference. This is about sexual orientation. It was from some of the same researchers on the previous slide, the ones that had done the twin studies. The primary author on this study is homosexual, and after his study was, he presented this study at this meeting, and some of these reports that I've read say he since left that lab because he was convinced, or he thought that people might have the potential to misuse the research he was doing if it carried on, because these sorts of news stories started to come out about this research project. Researchers say gene changes show who's gay. Oh, look at this one. Epigenetic tags linked to homosexuality in men. So this talk he gave was this author gave was all about epigenetic marks and whether or not those could help you figure out who was homosexual and who was not. gay or straight saliva test can predict sexual orientation. So I want you to be skeptical. So here's what these did. They collected saliva samples from 47 pairs of male twins. So it's another twin study. Probably some of the same twins that they studied in the earlier study that we talked about a couple of slides ago. And they looked at DNA methylation patterns on those DNA samples. And they looked across the entire genome, all the chromosomes, and they found that there were 9 regions of chromosomes where whether or not your genes were methylated or not was associated to some degree with whether or not you were homosexual or self-identified as homosexual. One of those 9 regions had an immune system gene in it. Another one had a gene that has some sort of neuronal function. It works in your brain. The other 7 regions, we don't know what the genes there do, really. Wah, wah, kind of a bummer study because you hope to find the silver bullet, like you find a gene that has an obvious implication on sexual orientation, but then these are the 9 regions of the genome you find. So associated 67% of the time means that if you look at these 9 regions, 2/3 of the time you will correctly guess, if you get somebody's DNA sample and look at these 9 regions and the methylation patterns, 2/3 of the time you'll correctly guess somebody's sexual orientation. That's what that number means. Why should you be skeptical about this research project? I have a number of questions. I hope you do too.
Why did they do it on twins specifically?
Right, so why twins?
Like, can they do it with just regular? No.
So why did they use twins? Same reason as before. They wanted to be able to know that they were looking at individuals with the same DNA sequences. But that would be an obvious next step. Let's take this out into the general public, not focus on twins anymore, and just say, hey, you on the street, spit into the cup. I'm going to tell you whether or not you're gay or straight. So that would be the obvious next thing to do, and I think, and that's one of the concerns is, do you want to have a genetic test? Even if it was possible, would you want to have a genetic test for sexual orientation? What other questions would you ask about this study design? Whenever you see a study design, anytime you see a detail, you should ask why that specific detail. So, collect saliva samples. What's the first question? Why saliva? Second question, why not do some heterosexuals as well? You'd like some control experiments, perhaps. So they did 10 homosexual, where are the heterosexual twin pairs? How old are they, and why would that matter? It's a good question. I don't know how old all of these twin pairs are.
They could have extra methylated or acylated.
Aha, so if there's an environmental effect, then maybe over time the environment changes those methylation patterns. So are you just looking at inheritance of methylation patterns, or are we looking at something like inheritance from the previous generation, or are we looking at something that's truly environmental?
Is this study's design part of that's in the previous library?
So this is a new study design, but it's the same authors. So presumably I assume, I haven't looked it up, some of these sets of twins are probably the same ones that they studied previously. I want to know why males. Why are all of these studies about male twins? I have no idea, but maybe I'll email the authors. Yeah, why males? Why not pairs of female twins? I don't know. I'm sure there's a good reason for it, but it's not explained very well in these media reports, right? Take the media reports with a grain of salt. Right? Be skeptical. So these are some of the concerns, just two to wrap up. Yeah. These are two of the concerns that have been raised by other geneticists as they've had access to the details of this story. One is, it's a pretty small number of individuals, so it's unclear yet whether or not that correlation between methylation marks in those 9 regions of the genome are just correlated, or if they're actually causative. Right, we've talked about correlation and causation a lot. Eating cheese and death by strangulation of bed sheets. This could be the same thing. It just happens by chance that in these twins, there's a random association of methylation patterns and parts of the chromosome with sexual orientation. So we need more people if we want to pursue this research further. And why saliva? Saliva comes from different cells early in development than other parts of our bodies. So who cares if your saliva is homosexual? Basically is the point. There are probably other parts of the body that are more important. If any part of the body is important in determining sexual orientation, it's probably not saliva. Maybe we can agree, I don't know. So maybe they need to get different DNA samples because their methylation marks or acetylation marks or whatever might be different. And who knows, maybe it's not DNA methylation, maybe it's histone acetylation that's important. So they're only looking at a tiny fraction of individuals, a tiny little part of the genome, and the possible ways that epigenetics can regulate gene expression. That should do us. So next time, we'll get more into regulation of genes, including the last case study.